Good afternoon, everyone. My name is Rod Ritz. I'm the co-founder and CEO of iVexSol, and I'd like to thank the organizers for the opportunity to speak to you about how our technology can solve the lentiviral vector crisis by harnessing stable producer cell lines and accelerating patient access for these critical and life-saving therapies. For those of you who don't know me, I've been in the field for about 25 years, first as an academic, and then joining in the early days of Pfizer Regenerative Medicine, one of the first pharmaceutical companies to dip their toe in cell and gene therapies. Then moved on to Novartis, supporting the work transferring the technology from pen to a commercial product, Kymriah, working in the technical research and development department, and then later in innovation. It was really during our time at Novartis that we first saw that there was an issue with getting lentiviral vector. We were looking to produce some 300 doses, and even then, we couldn't produce and didn't have enough vector. So shortly thereafter, I left Novartis and with my co-founder started iVexSol in 2018. We manufactured our facility in 2020 and launched in 2022. And what I'm going to show you now is some of our technology. So the field is really at an inflection point. You've seen that there's tremendous promise and inspiring successes with cases such as Emily Whitehead and others that are successful treatments. You see on her banner there that she lists that she has a cure, and we've seen these cures, across the board. Moving from pediatric ALL, the first indication, onto other indications, DLBCL, follicular lymphoma, etc. We're seeing success on success, and we see that CAR T demand will continue to skyrocket. We see that with earlier lines of treatment on the right-hand side and label expansions, moving into autoimmune disorders, that it's a very promising and a very strong fourth pillar of medicine. However, we have sobering limitations. We see a picture of a patient who unfortunately did not receive treatment in time, and he is one of approximately 20% of patients on waiting lists who do not receive these treatments because they cannot get lentiviral vector. It's wholly unacceptable, and our mission is to be able to provide vector to the field and break this bottleneck. So one of the issues in the field that's causing this bottleneck is the manufacturing of lentiviral vectors is using outdated technology. You see on the far left-hand side, they use a third generation or 4 plasmid system. You had 3 packaging plasmids, GAG, POL, REV, and M, and then you add your gene of interest to those. Unfortunately, the integration of that genetic material is unstable in your parental line, and it means that you really get minimal vector production. Minimal vector production, around 3% of the cells at any one time are producing vector. You also have your cells actively trying to eject that genetic material, and if it does happen to produce vector, those materials that are produced are toxic. So you really have a non-optimal system in which to produce large amounts of material. If you look at the graph on the far right-hand side, the gray is showing the field expanding, and the yellow is showing that the distance between what is needed and what can be provided is getting larger and larger. So what I'd like to propose and show you our data on is our turnkey solution that can solve the lentiviral vector process. As we show when we list our attributes on the left-hand side, we have stable lentiviral vector producer cell lines from multiple gene of interests, GFP, green fluorescent protein, and CART19, Kymriah. We have a serum-free suspension system. We have long-term cell stability, passaging them over multiple passages. We also have an ingenious material that allows us to harvest multiple harvests and store our bulk vector for up to 5 to 7 days at 4 degrees, and then combine it together to make a mega batch from smaller batches. And finally, our secret sauce is the folks that work with us are leaders in the field, and we can tailor-make a upstream and downstream process that is associated with your gene of interest and give you vector at very high quality. We feel that this group of attributes and folks that we have allow us to deliver a trifecta of value. That is low cost, high quality, and ultimate scalability. When you see scalability, you want to think about speed as well. Most often you only get 1 or 2 of these attributes. We feel that our system gives you all 3 and can reduce your cost of goods by up to 80%. So how does it work? Our system is different from the transient system in some ways, and in other ways similar. We use a third-generation self-inactivating vector. It's a 4-plasmid system, and we also insert the gene of interest from our clients. But what's different and is transformative is we have an inducible expression of each of those elements, meaning we can turn the elements on once we've reached scale. That avoids the toxicity that is seen in a transient system. Second, it's a stable transfection. Once it's in, it stays in. That allows us to make monoclonal cell banks that are on the right-hand side of your gene of interest. And when you look at those monoclonal cell banks, you see that almost 100% of the cells are producing at any one time. Those 3 changes, inducible, stable, and a clonal cell bank, is the key to our technology and allows you to have a highly scalable, rapid, and reliable production of lentiviral vector for the life of your product. So I'm going to show some data now on our candidate selection process. If we look at the far left-hand side under banner number 1, we see that these are clones that have been generated in our initial process, which you've seen on the previous slide. The dots there are individual clones, and if you look on the y-axis, those are your titers. As you increase up the y-axis, you get higher titers. The top panel is looking at green fluorescent protein as a gene of interest. The lower panel is CART19 or Kymriah, and you see that they're similar and yet scattered. The titers that we're seeing for both of those is somewhere around 1.3 TUs times 10 to the 6 TUs per mil, which is unacceptable, but we want to improve those titers. Step 2 is the enhancement of the clones. We then take those initial clones and through a proprietary enhancement process can increase the productivity of those clones between 10 and 100 times to get more than a clinically relevant titer of 3.2 times 10 to the 7 TUs per mil. So that's our process in which to generate the clones. Now what we want to do is to characterize them and find the best clones. What do I mean by the best clones? Under banner 3, you see several lines. In a traditional transient process, the blue line is this high productivity, and then a very short tail that says it bursts. It produces vector for a short period of time, and then stops production. The red is equally unattractive in that you never get the optimal vector production. What we're looking for in our clones is a green line where we get a high sustained production over multiple days so that we can harvest over multiple days. The second characteristic that we're looking for is a high titer and a PPTU ratio, high infectivity. You see our success criteria is that we want at least 1 times 10 to the 7 transduction units per mil, and we want to make sure that our infectivity is at least 1 in 5,000. Once we see that, we move on to process step number 4 and 5, where we look at process intensification, making sure that we can harvest 30 to 40% of the vector that's being produced, and then we move on to our stability studies. The next slide shows our advancements in stability. And here we're talking about 2 different types of stability. The first is cell line stability. We want to make sure that our cells, our stable cells, have the ability to produce over a long period of time. And you see that we have at least 90 days stability from our stable vectors. Why that's important is this allows you to seed a hollow fiber reactor or will allow you to seed a 200-liter or a very large reactor if you would like to produce a lot of vector. However, our second advancement avoids the use of these large bioreactors, and that's what I've told you about before, our stable solution that allows us to harvest material and store it at 4 degrees over 5 to 7 days. So in the schematic, you can see flasks that are there. There's 5 flasks, and what that's supposed to represent is 5 individual harvests from a growing bioreactor. Each one of those harvests is somewhere around 2 times 10 to the 7 transduction units per mil. So if we equate that to a 10-liter reactor, we see that if we were to harvest once, we would have 2 times 10 to the 11 TUs in total. However, having the ability to harvest 5 times multiplies that by 5 and gives us from that same 10-liter reactor 1 times 10 to the 12 TUs. What does that mean? It means from a 10-liter reactor, you can produce over 2,000 CAR-T doses. I'll say that again, 10-liter reactor, 2,000 CAR-T doses. That's a significant increase. So when we compare the two systems now, you make a choice. Would you rather hurdle your way to high vector production, or would you rather sprint? And what I've shown you here is that in the lower panel, by having stable integration and having the majority of our cells producing vector at any one time, and an inducible system remove toxicity, we can bolt to the finish line rather than having to clear higher and higher hurdles, including toxicity that's knocking out many of the cells in your culture system. And by doing that, we can produce 5 to 25 times more vector than a traditional system, and essentially produce vector on demand. That is, vector within 6 to 8 weeks of thawing your vial. So now let's switch and talk about another challenge in the field, and that is the high cost of goods and difficult scaling. If we look at a 50-liter reactor, our system has the ability to produce over 500 more doses from a 50-liter reactor as compared to a transient system. However, its real value, and I mean value from a dollars and cents point of view, is a reduction in direct cost of goods and indirect cost of goods. I've told you that this is a plasmid-free system. That represents approximately 50% or more of your cost of goods when you're producing transiently. We don't use plasmids except in the very first step, so you have that major cost reduction. The second is the indirect cost of goods. We don't need large reactors, we don't need hoteling costs and other very expensive, time-consuming steps that raise your indirect cost of goods. I've told you already that this increases the quality, reliability, and safety of our system, and that a master cell bank enables us to produce vector on demand, and what we feel creates for the first time the potential for an unconstrained lentiviral vector supply. You see here our investors and our partners. We're very pleased to have the support of those and to demonstrate data with some of these partners. And finally, the most important slide. I told you at the beginning that it was an unacceptable situation that we have this transformative technology that is not reaching the patients, and it's not allowing us to produce and capture all of the patients that we're looking to. iVexSol's mission is to improve patients' lives by developing transformative vector production technologies that will stop the situation where folks are not receiving life-saving therapies due to a raw material rather than the approval of a therapy. I thank you all for your attention. I too have finished early, so if there was any questions, I can take questions, or I can meet you at the end of the session. Yes?
First of all, congratulations. I've seen iVexSol from the start, so this is great. You're really addressing an unmet need, and that's great. Can you go to slide 7, please?
I hope so. It's not allowing me to reverse the slide. Oh. There it is.
Yeah, so the PE ratio, you're targeting between 1 to 5,000, right?
Correct.
What's the maximum you've seen?
So in an unoptimized, maximum is in bad or maximum is in good? 1 in 500 is the maximum that we have seen. The industry standard is about 1 in 5,000. It really depends not on the molecular biology, but more on the culture conditions. So you can take the same line and get a very poor PPTU ratio, or you can have a very high PPTU ratio, depending on how it's cultured.
Have you done the calculation to actually see what parameters are the most influential on that ratio?
That's still to be done. It also depends on the gene of interest. So the two gene of interests that we have there are small and relatively straightforward. We're testing more complicated and more difficult genes of interest to make sure that our system has the variability and the bandwidth in order to accept these more difficult situations. Great questions, though. I guess I should add, too, that our system uses a universal cassette. What that means is that if you produce plasmid in another supplier and your backbone has been dictated by that supplier, we can use that backbone. We can have universal backbones, universal genes of interest. So if you worked with someone else in a transient system and you want to come to us, we can also produce using those plasmids. All right. Thank you very much for your attention.